Hi students, welcome back to online lectures. Coming to enzymes involved in replication, part 2. Structure of DNA polymerase. Coming to subunits of this enzyme, it has 5' to 3' polymerizing activity. This is achieved by alpha subunit. And epsilon subunit has 3' to 5' exonuclease activity and plays a key role to ensure the replication fidelity. Theta subunit maintain heterodimeric structure. So alpha, epsilon and theta subunits together comprises 4 enzymes of the DNA polymerase 3. Coming to various subunits of DNA polymerase 3 and their functions. You can see here theta subunit encoded by whole E gene stimulates the epsilon proofreading act. Epsilon subunit encoded by DNA Q gene has 3' to 5' exonuclease activity. Alpha subunit encoded by DNA E gene has the polymerase activity. And tau subunits encoded by DNA X which acts to dimerize two of the core enzymes which are alpha, epsilon and theta subunits. Gamma subunits encoded by DNA X which acts as the clamp loader. Delta subunit encoded by whole A which acts as clamp opener. Delta prime encoded by whole B which acts as a clamp loader. Zeta subunit encoded by whole C which helps in interaction with single strand binding proteins. And Psi subunit encoded by whole D which helps in the interaction with gamma and zeta. Eukaryotic DNA polymerases. There are alpha polymerase, beta polymerase and gamma polymerases, delta polymerases and epsilon polymerases. See the functions of DNA polymerase alpha which has polymerizing activity of 5' to 3' direction involved in replication and in DNA repair. There is no exonuclease activity for DNA polymerase alpha. And coming to DNA polymerase beta which is involved in DNA repair and no exonuclease activity formed. DNA polymerase gamma replication in mitochondria 3' to 5' exonuclease activity. DNA polymerase delta involved in replication and DNA repair have 3' to 5' exonuclease activity. DNA polymerase epsilon involved in replication and have 3' to 5' polymerizing activity. Coming to a DNA clamp. A DNA clamp also known as a sliding clamp, a protein fold that serves as a processivity promoting factor in DNA replication. Especially DNA clamp proteins are beta proteins, beta subunits of the DNA polymerase 3. As a critical component of the DNA polymerase 3 holoenzyme, the clamp protein binds DNA polymerase and prevents this enzyme from dissociating from the template strand. The clamp polymerase protein-protein interactions are stronger and more specific than the direct interaction between the polymerase and the template strand. Coming to single-strand binding proteins, single-strand binding proteins prevent re-annealing and to protect the single-strand DNA from being digested by nucleases. Here see the functions. One is prevents the re-annealing, another one is protection from the nucleases and prevents secondary structure formation. And these do not allow the formation of secondary structures of the single-stranded DNA, thereby allowing other enzymes to function effectively on the single strand. Molecular weight of the single strand binding protein is 75,600 daltons. It contains 4 identical subunits which binds single strand DNA. You can see here, this is the double helical DNA where topoisomerase unwinds the supercoils and helicases separates the strand and separated strands are bounded by single strand binding proteins and the another enzyme which is involved in the initiation of the replication, that is PRIMASE enzyme, which reads the template strand to synthesize the DNA in 5' to 3' direction, followed by DNA polymerase 3 involves in the extension of the DNA strand using the 3' hydroxyl group of the RNA primer. So coming to the topoisomerases, every cell has enzymes that increase or decrease the extent of DNA unwinding are called topoisomerases. So they increase or decreases the unwinding of the DNA. Topoisomerases are also known as DNA gyrase and that act on the topology of the DNA. So what is the function of topoisomerases? They bind to double-stranded DNA and cut the phosphate backbone of either one or both the strands. So they bind to the DNA and cut the single strand or double strand phosphodiester bonds on the DNA. This intermediate breaks allows the DNA to be untangled or unbound. So this allows the unwinding of the DNA, and at the end of this process the DNA backbone is resealed again. Again the digested backbone of the sugar phosphate backbone again resealed. Topoisomerase is an enzyme that can change the linking number. Linking number is nothing but number of times DNA undergoes coiling. There are two types of the isomerases. They are topoisomerase 1 and topoisomerase 2. Coming to type 1 isomerases, this acts by transiently break one of the two DNA strands. So this enzyme acts on the one of the two DNA strands, rotating one of the ends about the unbroken strands, and rejoin the broken ends. They change the linking number in increments of 1. That means it removes the one supercoil at a time. Coming to topoisomerase 2, this enzyme breaks both DNA strands and change linking number in increments of 2. That means it can cut the both strands of the DNA and removes two supercoils at a time. Coming to topoisomerase 1, best characterized in Escherichia coli, enzyme is A64 amino acid long, monomeric, encoded by TOP-A gene. Mechanism of catalysis involves formation of covalent residue between the tyrosine residue and the phosphodiester backbone. The first one is enzyme must interact with the phosphodiester backbone. So here the enzyme linkings are covalent linkings, and especially the tyrosine moiety of the enzyme is involved here. The nucleophilic attack from hydroxyl group of tyrosine to a phosphorus atom creates phosphodiester link. So this link is the enzyme forms link with the phosphodiester backbone with the tyrosine. Tyrosine have hydroxyl group where it attacks the phosphorus group and creates the linkage between the phosphodiester bonds. This generates free hydroxyl group. See again, nucleophilic attack from hydroxyl group of tyrosine to a phosphorus atom creates a phosphodiester link between the enzyme and the DNA, generates free 5' hydroxyl group. The other strand is held in a place by binding non-covalently to a domain of the enzyme. The strand is again resealed. Thus one supercoil is removed. Topo 1 from E. coli acts only on negative supercoil. Eukaryotic Topo 1 can remove both the negative and positive supercoils. See, you can see here the enzyme cuts the single DNA molecule, and this allows the removal of one supercoil at a time. Coming to topoisomerase 2, best characterized in Escherichia coli, and this is known as DNA gyrase, is a tetrameric protein consisting of 2 A subunits and 2 beta subunits. Depending on DNA substrate, they can change positive supercoils to negative supercoils. So remember, this topoisomerase 2 changes the positive supercoils to negative. That means relaxed form of the DNA. Number of coils are less in negative supercoil, or increase the number of negative supercoils by 2. Topoisomerase 2 catalyze the catenation and decatenation, that is the linking and unlinking of two DNA templates. The enzyme also introduces negative or positive supercoils at a near or the origin C site of the replication. It also removes positive supercoils formed ahead of replication. This is the function of topoisomerase 2, which allows the cutting of two DNA strands, and the second strand pierces the other strand, and thereby two DNA molecules are separated. The type 2 topoisomerase cut both strands of the DNA helix simultaneously. They use the hydrolysis of ATP. Unlike type 1 topoisomerase, these enzymes change the linking number of circular DNA by 2. Coming to DNA ligase is a specific type of enzyme, a ligase, that facilitates the joining of DNA strands together by catalyzing the formation of phosphodiester bond. DNA ligase is used in both DNA repair and DNA replication. The mechanism of DNA ligase is to form two covalent phosphodiester bonds between 3' hydroxyl ends of one nucleotide, that is known as acceptor, with the 5' phosphate end of another incoming nucleotide. ATP is required for the ligase reaction, which proceeds in 3 steps. One is adenylation, that is addition of AMP of a lysine residue in the active center of the enzyme. First one is adenylation of the enzyme. There a pyrophosphate is released in this reaction. Now the transfer of AMP, this AMP to the 5' phosphate of the so-called donor, that is incoming donor, formation of the pyrophosphate bond. Now the formation of the phosphodiester bond between the 5' phosphate of the donor and 3' hydroxyl of the acceptor. In such a way, the sealing occurs. Ligase enzyme that seals the nicks formed during the repair and during the removal of the primers. Coming to DNA primase, is an enzyme involved in the replication of DNA and is a type of RNA polymerase. Primase catalyzes the synthesis of a short RNA segment called a primary or a primer complementary to a single strand DNA template. Primase is of key importance in DNA replication because no known replicative DNA polymerases can initiate the synthesis of a DNA strand without an initial RNA or DNA primer. So, the DNA polymerase can only elongate the replication, but the initiation is achieved by DNA primase. This is DNA dependent RNA polymerase. After this elongation, the RNA piece is removed by a 5' to 3' exonuclease activity. As we know, the DNA polymerase 1 has exonuclease activity. This reads the DNA in 5' to 3' direction and removes the primer, RNA primer, and there that is refilled by the DNA and short nick formed during the rejoining of the newly synthesized DNA segment, and next segment is followed by ligase enzyme. See here, you can see the complete picture of the enzymes involved here. So topoisomerases unwinds the supercoils, helicase separates the strands, and two strands are bounded by single strand binding proteins. Whereas on the template, that is having the polarity of 3' to 5' direction, the synthesis starts in 5' to 3' direction by DNA primase enzyme. Whereas coming to another strand, the direction you see the template available is 5' to 3'. So this is discontinuously available strand to synthesize these primers. So the synthesis of the primer here is 5' to 3' direction on the opposite complementary strand. You can see different functions here. See, topoisomerases mix and unwinds strands to release mechanical stress and unwinding. And next you see the helicase function that unwinds DNA at the replication fork. And now single strand binding proteins which binds to the separated strands and stabilize the single strand DNA at replication fork. Now RNA primase initiates the new strand synthesis in 5' to 3' direction. Now this can be extended by the binding of DNA polymerase 3 enzyme. So here the synthesis of one is continuous strand synthesis, another one is discontinuous strand synthesis. Why? Because here the template available for the synthesis is in opposite polarity. So thereby on this to synthesize the new DNA strand, so a short segments of the primers are required and discontinuous DNA synthesis is elongated. Thank you.